Hello all, welcome back. Today we are doing staining methods. So the first thing I'll show you how to do is a bacterial smear. You need to do this in order to be able to stain your microorganisms. So I'm going to show you for one, and then I'll come back to you as soon as I finish all of them. So basically what you do is you take a drop of DI water, and you place it on the microscope slide. So I'm going to place a drop on each microscope slide. And then what you do is you emulsify the microorganism in the droplet. Now with my Bunsen burner on and my loop straight, I'm going to proceed to grab the organism. I'm going to heat the loop, maintain my aseptic procedure, right? So make sure it gets red hot. Flame part of the handle because it's going in the tube. Remove the cap. Flame the tube. Go into the tube. I can cool my loop if I want on the top, and then go grab a little of the organism. So flame the tube, cap it, and set it aside. Okay, so now we have the organism on the loop. So now we go back to the drop of water that we put on the slide, and we emulsify the organism in the droplet. So we're just basically suspending the microorganism in the drop of water that we put on the slide. Okay, so I did this. So now I'm going to go back to the Bunsen burner, reflame my loop. And you don't have to see this, but just trust me, it's happening. And once I flame my loop, I'm going to place my loop down, and I'm going to return to my slide. So what you need to do now is you need to allow it to air dry, and then once it's air dried, you'll heat fix it using the Bunsen burner. So I'm going to skip ahead though, and I'm going to heat fix it. I'm going to dry it and heat fix it using the Bunsen burner. Okay, so here I'm taking the slide over, and I'll hold the slide off to the side of the Bunsen burner, and I'll kind of just swirl it. And what you can do is, since the bottom part of the flame is the coolest part, as you know, it's still hot, but it's not as hot. So you can pass it through the bottom part of the flame. And get the slide a little warm. And then you can keep kind of like moving the slide around. So as you do this, you'll notice something. So the drop of water, the way a droplet dries is it dries from the outside in, right? And that makes sense because the drop is thickest in the middle. So as it does that, as it dries from the outside in, it begins to pull any organism that's on the outside, the periphery of the droplet, toward the center. So if it does this, where do you expect most of the microorganism to be? That's right, in the middle of your specimen. So always, when you're looking at microorganisms on a microscope slide, you always want to focus on the outer periphery, because that's where you'll see less density, and you'll be able to determine the morphology of the organism, because you'll be able to see individual bacterial cells versus just clumps, like a big clump of cells. So, so you can see that the droplet's getting smaller. It's retracting to the center of the specimen. it continuously does this. So you can kind of hold it off to the side and just tip it. The slide is hot enough to where it'll, and depending on how much you put organism on the slide, will determine how much you'll end up seeing. But you should see this type of like haze on your slide. That means that the organism is there. If you don't see that, then you most likely didn't put enough, and it's going to make it that much harder to see when you go to focus it under the microscope. Okay, so we just keep doing this until it dries completely. So you just keep passing it through the flame until that happens. Okay, so we'll get back to this in a second. Okay, welcome back. So we've passed this through the flame multiple times, and now there's no liquid left on the slide. So now we can say that this organism is heat fixed to this slide. The reason why we do a heat fixing is because if we did not do heat fixing, then the organism would simply just wash off the slide when we tried to stain it or whatnot. So this ensures that it's fixed to the slide. Okay, so now what we're going to do is we're going to do the first part of the exercise, which is the simple stain. So as the name rightfully states, it's a simple stain, right? And so it requires only one basic stain, right? So right now I've chosen to use for E. coli methylene blue. So I'm going to take the methylene blue and I'm going to apply it to the spot where the specimen is, right? I don't have to put it all over the entire slide. I just need to do it where the specimen is. And then I leave it for the amount of time that's stated in your manual. So for methylene blue, it says for 2 minutes. So let's time it for 2 minutes starting now. So I'll come back to you in 2 minutes. Okay, so 2 minutes has passed, and now we're ready to rinse off the stain. So what we want to do in our stain bin is we want to take our clothespin, hold the slide, tip the slide down into the bin, pour off any of the stain that's there, and then using the DI water bottle, you're going to spray the slide. And you want to just spray it until you get most of the blue coloring off the slide. And you'll see blue develop on the base of the slide. So once you don't see any blue on the bottom, then you should be good. Okay, so we've more or less rinsed that off fairly well, right? So now let's go to the next step. So the next step is to actually dry the slide. So the way we do that is we use bibless paper. So don't confuse this with the lens paper that you use to clean your microscope. Bibless paper is much coarser, and you don't tear out the pages from this booklet, okay? So basically to use this, all you do is you dial it to a page, you place your slide in, and then you lightly, I can't reinforce this enough, lightly tap it, and then flip it to the next page once you've taken it out, flip it to the next page, put it in, lightly tap it, and just keep doing that over and over again. And this is, excuse me, this is simply just blotting paper, right? So we're just doing it over and over again until it's fully dry. Now you're going to use the same procedure to do the rest of your wet mounts. So I'll do those for you, and I will be back with you in a minute. As you can see from the slides in front of you, all microscope slides have now been inoculated with the organism and have been prepared. A bacterial smear has been prepared and has been heat fixed. So now they're ready to be stained. I'm going to continue doing this for the remainder of the specimens. And once I have them done, I'll be back with you and we'll look at these under the microscope. Okay, so as you can see here, this is the E. coli slide. In the E. coli, I used methylene blue to stain it. So as you can see, the shape of the E. coli is what? It's rod shaped, right? And you can see that the size of the rods is not very consistent. Like some are short, some are long, and whatnot. So we refer to this as being pleomorphic because it can take on many shapes. But for the most part, they're all rods. They're not. There's no cosi here. Okay, so now here we're looking at another organism. So this organism is Micrococcus luteus, and we stained it with safranin. What color is this, and what shape? That's right. It's reddish. It's like a reddish color, and they're cosi. And you can see here, in this spot, you can see that they kind of form tetrads. Tetrads are groupings of 4, and you can see it in the center of this image. So that's M. luteus. This organism here is Bacillus cereus. And you remember this from the first day when we did Microscope 101. As you can see from this image, you see the vegetative cells, and then you see in some of them a clear area. You can see it on the left-hand side here. You see clear areas in there. These are the endospores. And so the actual spores, you see, some are all already liberated into the surrounding area. There's no vegetative cell around them. So what color is this and what shape? That's right. B. cereus is rod shaped, and the color for this is violet. Violet. Okay, so the final basic stain that we did was of Staph epidermidis. This organism, as you can see, grows in grape-like clusters. That's a signature pattern of the genus Staphylococcus. So you can see that the shape of the individual Staph epidermidis bacterial cells are what shape? That's right, they're cosi. And the coloration of this is reddish pink, reddish pink. So that's this was stained with the basic stained safranin. Okay, next we'll move on to the gram staining. The gram staining procedure is done the same way as the basic stain. You prepare a bacterial smear, you heat fix it, and once it's heat fixed, then you can proceed with the gram stain. Remember, the gram staining technique is unlike the simple stain. The simple stain simply stains the bacteria with whatever stain you pick, and it colors all bacteria the same color. The differential stain will stain gram-positive organisms differently than gram-negative organisms. And that'll be described to you in your lecture. I will now conduct the gram stain, and I will be back with you after I finish the procedure, and we'll look at it under the microscope. So as you can see here, at 100x, the E. coli is very faint. And it's a pinkish, like a pink color, due to the safranin. the counter stain. So that indicates that this organism is what? Is it gram-positive or gram-negative? Gram-negative, yes. Okay, next we'll look at the Staph epidermidis. Okay, so here now you can see that I gram-stained the Staph epidermidis, and the staph appears to be what color? Like a violet color, correct? And you can still see the grape-like clusters of the staph, and yeah, so this indicates that this organism is gram positive, right? Okay, so gram negative, it's due to the thinness of which layer? The peptidoglycan layer. Okay, great. So the next slide we'll look at is E. coli and staph combined into one sample. So I'll be back with you in a second. Okay, so now as you can see, we have a combination of the two. So we have gram-negative rods that appear around, and we have a series of gram-positive foci that are in clusters. So, so this is a mixed culture would look like this, and then the pure cultures would look like the ones I just showed you. So, so when you're working on like an unknown and you're trying to streak it out and get separation and all that, if you were to do a gram stain of your initial culture tube, you would see this. You'd see a mix of two organisms. Okay, so that's the end of the staining methods lab. The actual streaking of the plate we will do actually now. So I will pause and get back to you in a second. Okay, so here we go. So we're going to do a 4-quadrant streak of a mixed culture of E. coli and staph simulans. So I'm going to take the organism, flame the tube, place it to the side. So before you take the organism, you want to kind of mix up the sample to make sure that it's suspended completely and mixed so that it didn't settle out or anything. Then go onto the plate and do your 4-quadrant streaking techniques. So for the first quadrant, I drew the guide on the bottom to help myself. I don't need it, but I mean, I just did it anyway. But with this, you'll be able to now see, unlike the plates that we did the other day, you'll actually see separation. Like you'll see two different colonies that appear on the plate. And they may look different, they may not, and so that's why it's important that you do the gram stain. So you'll take a colony, you'll gram stain it. If it looks pure, then you can transfer it to a slant and you can do further testing on it. But if not, then you have to streak it again, and you just keep doing it until you weed out the other organism. Cool, remember cool on the side. Then you're gonna do your streak across. So that's quadrant 3. And now we're on the last quadrant. It's quadrant 4. So we'll cool it right here at quadrant 4. And then once we've cooled it, we'll pull from 3 and then just taper to the center. And there you go. So we'll incubate this, and I'll check on this in a day or so. And that's it. And that's exercise 3, staining methods. This was meant to give you practice before the unknown in streaking. So that's the reason why we did this in staining methods, just to let you know. Okay.